Hi everyone, this is your biology class. I'm your teacher Seema Atreja. Student, in this video we are going to discuss the gel electrophoresis technique, which is used for separating DNA fragments based on their size. Gel electrophoresis, it is a laboratory method which is used to separate mixture of DNA, RNA or proteins according to their molecular size. In this method, the molecules to be separated are pushed by an electric field through a gel that contains small pores. Firstly, the sample DNA is cut into fragments by using restriction endonuclease enzyme. This is a agarose gel plate. On this plate, we use the agarose gel as a matrix. Agarose is a natural linear polymer which is extracted from seaweeds. We apply this gel on this plate and produce a agarose gel plate. On this we create these wells, and inside these wells we apply the DNA sample after cutting by restriction endonuclease enzyme. We also create electric field here, which consists of two poles. One side it contains the negative charge and the other side it contains the positive charge. As we all know, DNA fragments are negatively charged. They can be separated by forcing them to move towards the anode under this electric field through a gel matrix. The name of the technique is gel electrophoresis. Gel which is used as a matrix is the agarose gel, which is a natural linear polymer extracted from a seaweeds. The other term which is used in this technique is electrophoresis. What is electrophoresis? That is migration or separation of charged particle like DNA under the influence of electric field. So in the form of gel, we are using agarose gel, and here we creating the electric field. On one side is present the negative charge and the other side is present the positive charge. So charged particle DNA, which consists of the negative charge, we apply or put the sample DNA into these wells. So these charged particle DNA migrate through this gel agarose matrix under the influence of an electric field towards the anode. Agarose gel provides the sieving effect here. The smaller fragment of the DNA move farther and the bigger one is settled down near the wells. These DNA fragments are visualized when we apply the stain, that is ethidium bromide, followed by exposure to UV radiation. If we do not use this stain, we cannot see the DNA fragments. So we apply the ethidium bromide stain followed by exposure to UV radiation. Now the DNA fragment are seen as orange colored band. Orange colored bands are visible here. The smallest DNA fragments move farther towards the anode, and the larger fragments, they remain near the wells. Now these smaller DNA fragments, which are the separated bands of DNA, are cut out and extracted through the gel piece. This step is called the elution. Where these fragments are used, these purified DNA fragments are used to form recombinant DNA. These are the desired gene. We isolate them from the selected cell. It may be plant cell or animal cell or fungal cell or maybe any microbial cell. Then we join this with plasmid and forms the recombinant DNA. Later on, we put this recombinant DNA inside the host cell to replicate. This is all about in this video. I think the concept of gel electrophoresis technique is very much clear to you all. Thank you so much.